Yes. Yes, there will be list and yeah.
Okay.
Okay, so we can maybe start, and during my introduction, there will be a chance for other participants to connect. Good evening and welcome to our public health webinar. My name is Michael Grivna. I'm professor and director of the Institute of Public Health at the College of Medicine and Health Sciences in United Arab Emirates University. This webinar is part of the global campaign, This is Public Health. We are also proud to be affiliated with ASFER, which is Association of Schools of Public Health in European region. We are organizing these webinars monthly with the aim of bringing interesting speakers from different areas in order to highlight different important public health issues. Today, in this webinar, you will be able to ask questions. You can do so by clicking on the QA option or on the chat in the middle bottom of your screen. You can also submit your question during the presentation. We'll try to cover questions in the end, depending on the time. Now, allow me to introduce our speaker today. We are really honored that Professor Vasilis Vasiliou accepted our invitation. Professor Vasiliou is the Barton Chair and Susan Dwight Bliss Professor of Epidemiology, Environmental Health Sciences, and of Ophthalmology and Visual Science, and of Environment. He's chair of the Department of Environmental Health Sciences at Yale University in USA. Professor Vasilis received his Bachelor of Science degree in chemistry in 1983 and PhD in biochemical pharmacology from the University of Ioannina in Greece. He was then trained in gene-environment interactions, molecular toxicology, and pharmacogenetics at the Department of Environmental Health in the College of Medicine at the University of Cincinnati. In July 2014, he joined the faculty of Yale University. In addition to his funded NIH research portfolio, he's also director of the NIAAA-funded R24 Research Center for mouse models and metabolomics tools to investigate alcohol metabolism and tissue injury. Professor Vasiliou has published over 200 papers and edited 3 books on alcohol and cancer. Professor Vasiliou is the editor of Human Genomics and serves on the editorial boards of several toxicology and visual sciences journals. Title of his presentation today is Genomics and Translational Epidemiology as Drivers for Public Health. Professor Vasiliou, the virtual floor is yours. Welcome.
Thank you very much. And let me start sharing my screen. But before, yes. I would like to thank you so much for the kind invitation. Your university is in my heart. I'm Greek, and we're very close in cultures and everything. But I have visited, I think it was 3 years ago, and I gave a talk for your center, which we will mention that today. And also, I'm very honored to give this presentation today to you, which we would like to discuss, you know, the future of public health and the future of tools and how we envision translational epidemiology, how we take advantage of the genomic advances that we have in order to improve public health, and improve public health not only just in prevention, but also bringing the wellness to our population. So let me start sharing my screen. And you can see my screen, correct?
Yes, yes, we can see your screen.
Okay, let me see something. All right. Okay, so I have to close a few things here. So, oops, I'm sorry. What happened?
We are seeing only blue screen. Okay, can you share it again?
Let me see. Okay, can you see it?
Fine, yes, yeah.
Okay, so just to re-say again my steps throughout. So I got my chemistry degree from University of Ioannina, and then I did my PhD in the Department of Pharmacology with Professor Marios Marcellos, and I studied then the use of aldehyde dehydrogenases as a marker of exposure to chemical carcinogenesis. After that, I did a postdoctoral fellowship at the University of Cincinnati, working with one of the P450s guru, Daniel Niebert, and I worked with him on gene-environment interactions and mostly on the enzymes called aldehyde dehydrogenases, and of course cytochrome P450s and glutathione. And then in 1996, I moved to University of Colorado, where I became the director of the Environmental Health Sciences and Toxicology program, and also was member of the Cancer Center, and of course, member of the School of Public Health. So throughout my career, I have been working with the gene-environment interactions, specifically with these enzymes and antioxidants and so on. In 2014, I moved to Yale, where I became the chair, and I'm still working on gene-environment interaction, obesity, and diabetes, and neurodegenerative disease. Unfortunately, I don't have time to go through a lot of research today. I've chosen to give you some important aspects of where the public health is moving towards and what it is. So when I came here, I just wanted to make a photo representing my department, and this was done in early, like 2015, where we created. You can see here we have the environment and we have the humans, and at that time COVID was not existing, so you can see that we had the virus in there, which actually right now I'm debating, trying to get something into Nature if viruses should be considered also as environmental insult. Anyway, so what my department does, and in connection with all the other departments, is looking at the environmental insults, which could be from drugs, radiation, viruses, smoking, and so on, and food and diet, of course, and various contamination levels. So we're not only just the chimney when we're talking about environmental health, and when I accepted the position to come to Yale, it was because I wanted to integrate the sciences in order to advance public health. And I will tell you exactly what my aspects are. So the outline of this, and I'm going to try to go fast so we can have some time for discussion. We're going to introduce at the beginning the term of translational epidemiology with the current concepts and what it's moving towards. Then we're going to talk about genomics, and we're going to talk about genomics-informed pathogen surveillance, which was actually a talk that I gave, as I said, that was 3 or 4 years ago in UAE. Then we're going to talk a little bit about the importance of shared data with, taking advantage of the COVID-19. I'm not going to go to great details on that. I'm just using it as an example. Then we're going to talk a little bit about genomics and genetics in obesity, because it's very important in our days, and also in cancer prevention. But one of the things that I really like to introduce today is the interplay between the genome and the societies, and especially on social behavioral sciences. And I think this is a new era, and I'm going to introduce also the clinical neuroscience, the concept of environmental neuroscience as well. Anyway, and then I'm gonna try to give you a few examples of integrated omics and deep learning, which essentially I think this is the way that we need to go. I will mention that through the lecture, and then we're going to talk about thoughts about the future of public health, and that's why I would like to open the discussion. So what is translational epidemiology? It's the study of risk factors and disease in large population. And now we have the advantage of using big data, electronic health records, that we can really make a revolution into the studies. But anyway, the importance is to harness power of the modern phenotyping, including large-scale omic approaches and validation models. So as you can see here is we're going to try to incorporate this huge, you're not talking about 1,000, 5,000, 30,000. You're talking for millions of data that are going to be available. There are a lot of challenges, of course. We can discuss that. But initially, we're going to discuss about the genomics as the first frontier of translational epidemiology. Then we're going to talk about a few examples of functional biomolecules, because you have the metabolome, you have the epidemiology, you have the data, you can get the phenotypes, but then you're going to get kind of a mechanism to get some more information of the causality. And I think the causality lies to the functional biomolecules, and that includes the metabolites that you study through metabolomics, proteomics, and the proteins, which really play a role in metabolites, and the RNA and transcriptomics. Finally, we're going to discuss the exposures on the exposome and epigenetics. And finally, we have the omics, machine learning, the new stuff that going on right now in every country. Finally, one of the concepts that I would like to also discuss is a validation mechanism, and why we need a validation mechanism, because so far everybody is talking about epidemiology being just as indicating associations. We don't want that. We want to get to the next levels. Next level. And the next level is causality. We want to create legislation. We want to create new laws and prevention models that will enable us that. And to do that, we have to have a causality mechanism. You know, it's very difficult to go with association studies. As I said, we have to take advantage of our electronic health record that they have emerged as an important resource for population health and clinical research. There have been significant efforts to leverage the electronic health record data for research. However, we still have a lot of issues here. Given the data security concerns and the complexity of the data, EHR are frequently difficult to access and use for clinical studies. There are a lot of consortia that they try to bypass that. We're trying to generate, as you can see, open access data that people can go and do that. So we are on a good way to progress that. So I'm going to start with my first topic in here, which was a lecture, as I said, that I gave. It was 3 years ago. Actually, it's going almost 4 years now. Genomics informs real-time global pathogen surveillance system. And I want to tell you how, how this review published in 2018 in Nature of Genetics, how concrete it was and how predictable it was for the case of the COVID-19. So the whole idea is how we generate a surveillance for an outbreak, and then you have a response. And again, the genomic sequencing here is very important, and then we're going to digital epidemiology, and we go to the One Health. So essentially, you have people that they're infected, you take a swab, you're taking the DNA, and the idea of having the portable sequences is really what makes the life easier. So you get this sequencing immediately, and now is whole genome sequencing of those pathogens. And then you have a lot of things that to consider, but is the depth of coverage and so on. But we have advanced quite a bit on that. And then the power of informatics, you put this information, you assemble the whole genome, you compare with reference sequences, and then you have a confidence threshold of what you have identified. And then you have the action, which is interpretable report, clear clinical and epidemiological action. So this is the big way of responding to something that happens. And we did see that, and I will give you the few examples how it happened into the COVID. So inferring the transmission from genomic data and also the emergence of infectious disease. So essentially, remember, when we have an outbreak, you isolate, as I said, you isolate the pathogen, and you have a control isolate, you have the DNA sequencing, you identify the sequencing, you're mapping it to the total, to a reference genome, so you can see to what it is. The capabilities we have now with the open databases resources is amazing. So you can do that very fast. And back in the old days, it used to take days for the computers to process the data, but now it's a matter of I would say, minutes or hours. In addition to getting the sequence in here, you can also see the individual sequences. And in other words, you can see the variants. And as we're very well informed for COVID, there are quite a few syntheses. So the virus or the pathogens evolve, and they have mutations, and you can create a kind of phylogenetic tree of those variants. And then using the phylogeny inference, you can go to the epidemiological reconstruction, and then you can find out the transmission inference and so on. So right now, if we can go for the COVID, for example, we have that the Delta has been one of the virus that is, it can cause significant disease, but then you have the Omicron, which is more transmittable, but less, at least it's not as bad as Delta in terms of outcome of the disease. So here is, we're talking about how the outbreak, it occurs, but something in most of the cases, everything starts from the animals, I would say. Not everything, but it has a very high probability. And again, look at that in this one of 2018 publication, there was a bat in here, which was transmitting this to the domestic animals. And then from the animals, this is the pre-emergence, and we go to localized emergence in here, where the pathogen goes from the animals into the humans, and then from humans, it starts getting spread around. This is exactly what it was predicting in here, and we have been talking, the examples that I have given in that lecture was about Zika virus and the Ebola, and now we have the SARS-CoV-2, and of course the United Emirates know about the SARS virus, which was an outbreak in 2012, if I remember well. So anyway, this virus, this pathogen of the virus has been identified. It is believed that it started from the bats, which most likely went to the wild animals or domestic animals, and then eventually went to the humans and then has been transmitted. So this is the human transmission, and it has gone on and on. So again, back in that time, there were quite a few things that were very important. First of all, is just the model of surveillance, which really relies on genomic epidemiology in here. And essentially, the surveillance is you identify a hotspot, you go through the process in here, you identify what is the pathogen in here, and then you go for a local surveillance and rapid response activation immediately, but also you deposit in public accessible databases. Why I'm saying that? This is the whole story in here. This can go for global community analysis, but also it can be used by other organisms. So this review for me, it was really amazing for its time, which indicated and showed the path that we followed for the course of the COVID-19 epidemic. So the importance of data sharing and integration in real time is you need real world and non-traditional data solution. You need to develop networks in real time for data sharing. This is very important for public health, and leveraging existing research tools, but also extend to reach of the current resources. We always need to evolve. You know, we need to take advantage of the open accesses. You need to bring it up into the needs of the everyday's life and go over. So the genomic surveillance was very important for identifying and tracking the SARS-CoV-2. And essentially, this is the paper, the monumental paper in Nature in 2020, which have identified the coronavirus, that it was coming from this. And in this particular case, there were almost 2,000 cases reported that they have been the first hospitalized from the 2019. So through the epidemiological change and everything, they did a full RNA screen for this virus, and they have identified it on the 2019 nCoV, which essentially it was predicted to be the virus, the SARS-CoV-2. So I have included here this fantastic news and views from, again, from Nature. You can see genomics and epidemiological surveillance, which essentially confirms what I've been telling in the first couple of slides. Anyway, to make a long story short, and what it says in here, the timely detection of surveillance was indicated right here on the sequencing of that. But the other thing is that I want to draw your attention is, you can see everything in highlighting in here, and here there are connections that you can go on. You can see the consortia that they were constructed in order to really bypass this pandemic. And you're talking about databases and open data access for investigators throughout the globe that they could contribute in order to offer solutions for this pandemic. So this was a truly amazing stuff, and indicate the role of the genomics on and epidemiological surveillance. I'm going to switch gears now, and I'm going to go to an issue which is the genetics of obesity. We know that obesity, it has been about 4 times as high the last 30 or 40 years what it used to be. I mean, the increase is simply dramatic in almost every continent on Earth. And of course, we can go, we can talk about how this can happen, but it does happen. The idea is, what can we do as public health practitioners in order to improve and decrease the obesity as much as we can? Of course, you can say healthy diet and so on, but you know, sometimes it goes a little bit beyond that, and you have to really look at the genomic and genetic information. So two things that I would like to mention here is that you have the monogenic, which is an early onset and severe obesity, and it has this characteristic here of high genetic contribution, single mutation, large genetic effect, and so on. And you also have the common obesity that occurs to a large population in here, which is a polygenic. So monogenic are on your left-hand side in here. Can you see my cursor moving?
Yes, yes, we can see it. We can see it.
Perfect.
Yeah.
So you can see on your left-hand side here, and this is something that was just published. You can see on the left-hand side here, the genes that are offering the early onset of obesity. And on the right-hand side, you can see, the multiple effects that you have, genome-wide association link studies, and the cumulative loci, which really going to high numbers. And of course, it's a polygenic effect that we're trying to really address. So what I would like to point here is we have advanced in terms of the single genes or the multiple genes and the genome-wide association studies. However, what it says this review, and this is really important, is that post-GWAS, genome-wide association studies, is the cross-disciplinary collaborations which combine genomics technologies and analytical approaches that they have started to facilitate translation of genetic loci into the meaningful biology and new avenues for treatment. This is what I was telling you at the beginning, that we really need to go from just association studies to causality studies. And how we can do that? Utilizing the multi-omic approaches and then find the mechanism. And essentially what we're trying to do now is, okay, you have found some genes that they might be causing an effect. You need to create a model, either human cell lines or 3D organoids, or an animal model that you can show these mutations can cause this effect. And that's what you make a strong argument about the causality. So it's not only just the genomics, we need to integrate also other effects. So moving forward is the genomics into the global cancer and global cancer prevention. I just want to tell you, we have done, a lot of people have done a lot of studies that they have then identified a lot of loci either throughout GWASs or sequencing of either the parental tumors or in several occasions here. You can see that the loci that have been identified through the various cohorts is, for breast is from 200, and then you go down to the kidney, that there are 13 loci which have been identified. Again, the whole idea here, and this is very important, we're going to be talking about some genes that have been identified, and that like BRCA1 and BRCA2 or P53, which we know they pose an immediate threat for the cancer. The idea and the point that I want to bring here is, how do we handle this in terms of implementing into the public? How do we deal with that? So what I would like to include is, in addition to identify those loci, is how do we consider policies? So considering how risk prediction and risk evaluation and reduction interventions might be perceived in different populations and countries is worthwhile. So here is the example between the BRCA1 and BRCA2, between USA and Israel, and also you have here in Brazil, in some other countries. I'm not going to start saying here is the founder variants have been identified in both populations, but I want to take you in here in some cases which shows differential implementation. So favorable cost effectiveness has not been assessed into the to one part that has been assessed in the other, and also the availability of counseling and follow-up services, which is not here. So if you don't have that, and if somebody finds a mutation in your population and you don't have this counseling available, people are feared. They might not even go and do the genetic test because they don't know how to handle that. They want to avoid that. So you really need to bring all these things together. So the roles of implementation research are to understand the factors that they can influence the acceptability and preventive healthcare interventions, and to adapt and tailor strategies with community engaged participatory research in order to successfully implement evidence-informed guidelines. So we really need to be working with the public, and in order to implement strategies. Here is a population and health equity network for translation of genomic research into the cancer prevention. So on the 3 panels on the left, you can see that you have a knowledge generation, and then you have a journal, a knowledge integration, and then finally you have the information dissemination here. You can see health policy, clinical access, tailored to prevention, surveillance, high-risk population, education, and training. But essentially what it goes down to is the effect of population. Do you have equitable access to cancer genetic services across the population? This is very important. We need to offer genetic counseling and testing, new risk reduction and evaluation. And also for population health, new cancers averted, cancer deaths averted, and daily DALYs are gained. So it's very important how you incorporate this whole platform in here. And that shows again that multiple sectors have to be incorporated in order to improve the public health. And when I'm saying multiple sectors, we really need to have the health practitioners, but also we need to bring into the game the private sector. We need to give incentives into the sectors to work together in order to assess public health. I know time is running, but I am really excited about, the prospect of the crosstalk between the genome and society. And what I say, from genome to society and vice versa. There was a few back in December. It's a story from the Yale News, which is a paper from a colleague of ours. It's Ranjita Sinha, which shows that physiological and biological resilience modulate the effects of stress on epigenetic aging. This is a monumental paper, and there is another one that has been published we will discuss that indicates how the stress makes life clock to tick faster, and also indicates that chilling out slows down. So social behavioral stuff needs to be incorporated in order to access all these effects. So the whole idea about this paper is what they did is they modulate the changes in the effect of the social behavioral statuses and the stress into the related physiologic expansions. Can you hold on for a minute?
Yes, of course, take your time.
I'm sorry about that.
Oh, it's okay. It's okay.
All right.
No problem.
So, so essentially, the point that this paper makes here is that the received environment, it can accelerate aging, and it can accelerate aging through modifying your epigenome. So make changes, not mutational changes, but changes into the to your DNA that it can result to a poor health. I think this is monumental, especially for stress. And we always discuss, okay, stress can really hurt your health. Can harm your health, but this is it tells you how this can be done. The other thing which is very important here, and I want to tell you, is the integrating genomics and behavior contextual factors in health and illness. So social behavior and environmental exposures accumulate over the life course, and they are multiplex, adding to the complexity of the gene environment interplay that impacts the health. And I have a slide to show you the importance of what we call environmental neuroscience, which is really fascinating as well. So discovery research that identify genomic underpinnings of the social behavior and neural phenotypes is potential quite powerful as we imagine clinical and community translation of such discoveries through precise medicine. This is really important. As I said, it's a field that it develops so rapidly. And especially now, listen, it's not only just mental issues, but for example, the pandemic is creating so much stress, so much changes into the social behavioral stuff that if you combine it with environmental exposures, you can see the way that it does and how, you know, the genomic structures really can have an effect is really important. And this is what the science is going through right now in terms of the public health. So from this particular paper that it was, it's fascinating. And again, I have this and I'll provide you my PDF of the slides. You can go and read all this. So essentially genomic discovery informed by social behavioral research. It can go to the investigation of social behavioral factors into the genomic translation and vice versa. And then in here, you can also incorporate the health equity and the interplay between the these factors that they can go back and forth. So it's a circle that we really need to consider and need to be addressed for a lot of issues. And this is the promise or the premise of environmental neuroscience. And what I would like to tell you here is that, you know, we have been exposed to various factors. First of all, you have the indoor, the physical elements, the indoor and outdoor exposures. You know, you have your indoor environment, the outdoor environment, pollution and so on. But you also have the social elements and the behavior. So how all this get to the environmental neuroscience and how this combine through your DNA in here. It could be on a bad side, on a good side here. And then you can see the functional connectome is how you get all these synapses in your brain talking to each other and go and identify either epigenetic or genomic isolation. And this is something that is going on right now. And this is how, you know, it can be based. I wish I had more time to go over on that because we have, I've created an initiative here for looking at the environmental factors on autism. And then you can see an integration between genetics and epigenetics at levels of, you know, exposures, but also on the social behavioral science that you can incorporate. So the future is not just exposures. The future is not just the genomics. It's multiple factors that they can have. And now we have the tools and we have the power to study that. And this is what makes science so exciting. Anyway, just to finish up with the genomics, we have the basic genomic research, and we also have the clinical, what we call the clinical genomics, genomic learning healthcare system. So you can see you can go from a hypothesis to genomic technology and so on, genomic data and analysis. You can take it to the clinical practice in here. You can go to genomic medicine practice innovations, outcome data, and so on. And so this is the crosstalk between the basic genomic research and to the healthcare. Moving on forward is, I'm going to talk a little bit about what we discussed about the integration of various omics and in solving and trying to convert the association to a causality. So again, genome, epigenome, transcriptome, proteome, metabolome, and then you have the environment in here. You have changes, and you can, this essentially can interact all these factors, but you can study this, how you can really convert the healthy people to unhealthy people and so on, and how you can intervene. Very quick, I'm going to go through that. I'm going to start with metabolomics, And I wanted to show you have a huge project. We're writing up right now. When we look at the metabolomics of the COVID patients and trying to compare that with clinical data that we have the clinical characteristics. This is a big project with the Yale impact, but my lab has done all the metabolomics, and we're using artificial intelligence now to with Kirill Veselko from Imperial College to really leverage the information from the clinical and get that. So our cohort includes a lot of people, 300 people, and for metabolomics is quite a big number. We're using the untargeted metabolomic, and I'm going to go this is just a chromatograph. On the plasma, you can see every peak is a metabolite. And this is just analysis from 40 data, shows that we have found significant difference in several pathways, and we have several motifs that have been generated throughout this analysis in here. And now what we're trying to do is we're trying to incorporate this along with the clinical phenotype. So can the metabolites tell us about the outcome? And of course, this is based on the Delta variant, I presume, and then we have to move forward with, perhaps with the Omicron variant, which is Omicron, and see what are the differences. But comparing those big, large data sets, and for metabolomics, although it's just it's a few hundred samples, the amount of data that are generated is unbelievable, and especially when you try to go that. Anyway, let's go to the exposome, and I would like to define the exposome. The exposome is the total exposures that somebody has gone throughout the life, and from the conception also to the old age, and how this has effect. Actually, now what they're talking about the exposome, they try to expand it, even the preconception, which either father or mother have been exposed, have been exposed for that, and that's another issue. So I have I had a conference here at Yale in 2017 about lifetime exposures. You can go to this site, and there's still the recording of this really successful conference we have, and we plan to do that more. So here is another example. I'm going to try to speed up and get time for discussion. This is the exposome and genomics. This is how you can use large population. This is from Michael Snyder, who used to be here at Yale, now is in the Stanford. So essentially, you can see that you can have the exposometers, so you can have personal devices that you can measure the exposures that they are there, and then you can do the DNA and RNA and the sequencing analysis, and then you can see, you can really make the population and study analysis and find out, the putting into the environment cloud and look at the variation, decomposition, and then you can see at the species analysis and so on. But what I'm trying to pass here is that you can have the chemicals that are present in your environment along with your transcriptome. And now if you add to these studies now more also, if you go to the individuals and you measure what are the changes at the metabolome level, you can expand this science into the next level, which is the clinic, the integration. And I think I have shown you a few of these. So putting also, that brings the issue about the, brings the issue about the deep learning, how you can use the deep learning to get all this stuff and all this data. So putting the deep learning to work for public health, this is the long-term exposure of the low ambient air pollution concentration and mortality among 28 million people. This is results from 7 large European cohorts with the ELAPSE project. This is the initiatives that Europe is funding right now. And actually, we just participated in an application going forward on one of this as a part of but anyway, to make a long story short, from the number of cohorts in here, you can identify the level of confidence about the PM 2.5, the nitrogen dioxide, and the black carbon in terms of causality of non-accidental mortality analysis. This is how you can analyze the big data. This is, but you need to utilize these big computers. So the whole idea here, and this is what the concept that we're working on, this is artificial intelligence and machine learning in terms of make data-driven healthcare. So the idea is you get your lifestyle, diet, environment, genetics, and also the gut microbiome, and you generate biochemical, metabolic imaging, nutrition, medical, social, and other type of stuff, and you do the analysis in here, and this data analytics is capable now to integrate all this information and creates motifs, and that will tell you about nutrition and lifestyle, drugs and dietary, and the medical. So this is the personalized intervention or precise medicine intervention in here that you have to use it, and you go from stratification to personalization to optimization. What I'm trying to tell you here is how these algorithms can help you and how these computers can incorporate. You can train this. And if we go back to the slide, I don't want to go back to the slide that I have shown you. The connectome between the brain is the artificial intelligence, and intelligence is being based on the human brains and how this connects and absorb the information and can really modify it. So this is an amazing science that we really need to incorporate in terms of the public health. So I have a couple of slides just to show you. I'm not trying to impress you. What I want to tell you is the importance of this deep learning. So this paper published in Nature Communication very recently, it shows about the integration of machine learning and genome scale metabolic modeling that identifies multi-omics biomarkers for radiation resistance. So this is, you're taking the during the chemotherapy, you have a lot of tumors that they become resistant. The same thing with pathogen, the bacteria that they become resistant to antibiotics and so on. But the whole idea is how you can utilize this multi-omic approach. And excuse me, you have here the gene expression. The gene expression, you can incorporate this from a huge number of samples, and then you're taking the metabolic features in here, you incorporate the metabolism. What I want to say one more time is here is data and data and data, and then you go to finalize, again, you go the clinical and from the metabolites, and you have the non-invasive, but the whole idea here is you can create a predicted probability of radiation resistance. So what I'm trying to tell you, if we were go back in our old days of research, usually you had to test a hypothesis that if this gene can affect the gene expression, you had to do all the experiments by yourself, and you had to really drive and go utilize maps or everything that has been published, try to get this information. Right now, what the technology enables us is to really investigate hundreds and thousands, and in some cases, millions of samples, and then does the work back. Millions of people can do it together, and that will be impossible. To get what? To get a predicted probability for radiation resistance. And this can really help you in terms of the improving the public health. So the future of the public health is we need resilient, robust, and multidisciplinary research. We need big data and data sciences. It's very important. We need really to bring more scientists into this area in here. And, it's not anymore biostatistics. Our biostatistics departments, they have to be converted on the big data. They still are biostatistics, but essentially this is how to handle these big data sets. Also, we need to have shared data across in real time. And what I'm saying in real time, and you can go back to that commentary from Nature, and you can see what it had happened with the COVID-19. Also, you need partnerships working toward the public health. I mentioned that before. We need the private sector and the public sector and the federal regulators and also the community to participate in order to promote public health. And we also really need to pay attention to health equity. NIH actually right now, it gives a lot of attention to the health equity, and that will help us really solve the issues. Before I close, I would like to tell you that take advantage of this seminar. We have a summer international program here that I've developed at Yale, and we have used to be we had before COVID, we had I think were five students from UAE, which they have attended this when it was in person, and they loved it. And it was really a good exposure. But because of the COVID, we offer it now as a 2 weeks lectures, which is on climate and energy impacts and health, developmental origins of health disease, green chemistry solution, and understanding and overcoming environmental health disparities, and of course, the novel approaches, the omics approaches that we talk about it, and implication in major human disease, cancer and COVID. This is the web page. If you need more information, you have my email. This is a 2 week, there is a tuition in here, but you can do it online. And we have been having really good success in attracting the people. And there are a lot of lectures. You're gonna get a lot of information regarding Yale, applying to Yale, PhD program or MPH program. With that, I'm gonna stop sharing, and I'm going to start the discussion. Thank you so much.
Thank you, Vasilis. Thank you so much. It was a really fascinating presentation and a lot of very interesting information. I'm sure that there will be some questions related to it. Maybe I can start the discussion. It is really the genomic studies are incredibly important, and you showed it for the public health and for surveillance, and also especially related to the COVID. I would like to open a little bit with a question on the ethics. And you know, many times public is quite scared on the, you know, the manipulation of the genes and how it is perceived. So if you can comment, you know, about you know, ethical issues a little bit.
Well, there are quite a few ethical issues in here everywhere. You know, once you start talking about genetic information is how much you can go. But, you know, what we have to look at the importance is how we can utilize this genetic information unidentified without having the people giving up the personal information, and how we can get that and then offer that back to the public. And that's why on my slide, I said we need a good implementation processes that will take into account the ethics. Absolutely correct. And you can really not utilize that, because sometimes and of course you may have some people trying to take advantage of the genetic information and trying to develop some other tools which are not. But, you know, this is what the governments have to be taking into account, but it's a major concern. I completely agree. Have we solved on that? No, I don't think we have solved everything, but there are standard processes that have been implemented by WHO, which can be followed.
Great, thank you so much. We have a couple of the questions I will try to cover in the time what we have, at least a couple of them. There is a question about data-driven research. How do you see the challenges in getting really representative and good quality data, for example, in terms of ethnic, socioeconomic, and so on aspects?
Very good question. This is why we need international collaboration. This is why we need the international cohorts. This is why, you know, Europe has done a tremendous job in incorporating these big cohorts. Yes, you should have, you know, your cohort should not be restricted to a particular population, and you can have multiple cohorts to address that. So this is the international consortia, which they were applied for the COVID. I mean, look at the COVID. If you look at the COVID, you get your answers. Look what happened immediately in South Africa when we had the Omicron and the exploration of that, and how this immediately went to everywhere. You can do that, we can do that. This is our effort to to make this happen, and this is rather important. And I just posed the same question. Last week, there was the council of the NIHS, and I posed the question, is NIH, the US NIH, open to international collaborations or not? I'm not talking about for example, collaborations with India, or collaborations with China, or collaborations with, UAE. I'm talking about putting the big consortia that Europe was able to do that. In Europe, you can put consortia that could be involving various countries. And the director of the NIHS actually said that this was a great idea.
Excellent. So another question from Mr. Aminu Abdullahi. Thank you, Professor Vasilis, for such an educative presentation. Non-communicable diseases, including endocrine and metabolic disorders, have been linked to developmental origin, sort of early life. Recently, interventions to address such developmental origin has been tipped to be promising in reducing the burden of NCDs. How can genomics play a role in the search for suitable early life interventions to prevent these chronic conditions, and how can big data be used in this quest?
Very good question. So actually, this is what we're trying to address with this new center that we pose for autism. So the idea is you have environmental exposures. How the genomics? I'll give you a very simple idea, a very simple problem. Acetaminophen, right? Acetaminophen has been lately accused or implicated on the development of autism. And I'll take you back to the basic pharmacology. What is acetaminophen? How acetaminophen can create toxicity? It can create toxicity through cytochrome P450s, or maybe if it's not conjugated with phase 2 enzymes. So if you have genomic changes on these enzymes, they can make a person more susceptible or less susceptible to acetaminophen inducing autism. And this we can go to PFAS, we can go to polycyclic aromatic hydrocarbons, or we can go just to any regular disease. So there is an interplay. It's gene-environment interaction, which is really need to be into consideration. So you can have the epigenetic changes, but at the same time, you have to consider, this is what I'm talking about, the big data. And now whole genome sequencing is very cheap. You can incorporate all this data.
Dr. Balash is asking how external exposomes, the entirety of environmental exposures a person is targeted with, can be measured in an agnostic way.
Well, that's a very good point. Now, of course, you can say somebody has been exposed, and you might. So the whole idea now, the answer to that is, this is a very, very good point. So what we're trying to develop now, we're trying to develop models based on not only the patterns that you find on the exposome with the wearables, but also the changes that they occur to your blood throughout the years. And you can model the exposures. In other words, utilizing this amazing technology and identify, has these soldiers been in that war, had been exposed to this particular warfare, or have been exposed to any other disease. So you can model that. This is what we're trying to do. And this is the beauty of artificial intelligence and deep learning can provide to you.
Dr. Mahmoud Sheikh Hussain, he's asking, thanks, Professor, for the fascinating and complex issue. However, when is optimum moment for public health intervention along genomic research on emerging pathogens that require immediate intervention?
Yes, it does require immediate intervention. And I think, to the extent that you can. So let's go for the intervention. Let's go to talk about, the particular one with COVID, right? So the whole idea is, how do you deal with this pathogenous virus? How you can intervene with that? So the first intervention was just creating these vaccines that will help you, but how do you treat the disease? And this is why you need open access data. You really need to bring public sectors and private sectors to work together to speed up the intervention. Yes, I agree with that. This is absolutely correct. we need to improve on that, and you need to have that through the open access collaborations.
Another question, thank you for pointing out the social and behavioral science perspective and interventions. However, the implementation research is very much in the beginning. How could we increase emphasis and funding for this kind of research, like for most of the multidisciplinary research?
Well, that is correct. The implementation is an issue, and implementation from every aspect. The social, this is an area that we know that from the old times. However, right now it starts becoming into the mechanistic aspects. So we still need to bring more teams together to analyze the data and to get that. And funding, I think everybody right now, all the governments are very concerned about the mental illness, mental health of the population. So I predict that you're going to start seeing more and more funding from all governments throughout the world.
I think it is incredibly important issue, what you were mentioning about the translation of genomic research into prevention, into action. I think it is not only genomic research, but public health research to transfer to the prevention. I think it is a very challenging issue, of course. Another interesting question. Hi, congratulations on the lecture. My name is Lina, and I had the privilege to work with Native Americans in the USA. Through my work, I heard the term of epigenetics and its role in diabetes and alcoholism among Native Americans. Have you and other researchers used any information or lessons learned from the problem of diabetes and alcoholism and its relation of epigenetics among Native Americans? Also, how the social determinants of health, education, economics, access to health relate to genomics and move us forward towards promoting health? Thank you.
Well, that we can start discussing actually what she, right?
It is Lina, yes, it is she, yes.
Lina, you brought up a very important question. I have to tell you, I work with alcohol and alcoholism from day one in my research life. So the fact of, I can give you a small lecture on that. I don't care if we go a few minutes extra. So you know that the Asian people, they have a mutation of a particular enzyme, ALDH2, so they cannot metabolize acetaldehyde. So when they drink a little bit of alcohol, their face becomes red. This is called the alcohol flushing syndrome. This is a genetic mutation. This is the most well-studied genetic mutation, a single gene mutation. Actually, there are a few more in the alcohol dehydrogenase stuff, but this is the major thing. If you go to Native Americans, this is different. They get a red face, but they drink more. They are capable of keep drinking and drinking and drinking. So I had looked at that sequences back in my early days in Colorado, and there was no difference in the DNA sequence of this gene in there. So there are epigenetic mechanisms, and the epigenetic mechanisms, they can affect the metabolism of alcohol, but especially, for this, for Native Americans, this red phase that occurs to them is completely different, and it could be due to the responses to the alcohol and vasodilation, but it does not prevent them from continuous drinking. So the Asian people, they have half a glass of wine, they become red, they cannot drink more. If they force themselves to drink more, they're gonna die, because acetaldehyde gets to the high levels and can kill them. So yes, have the people been studying for Native Americans? Not as much, but they start doing that. Actually, many of these big projects that we have, either Superfund projects, they really, NIH is forcing us, not forcing us, encourages to use the tribal population into those studies in order of, in a matter of health equities.
And maybe last question before we'll close. It is from Dr. Azar. She was asking, what advices you can give us in UAE as public health? We are about to finish the Emirati reference genome.
About what?
About Emirati reference genome that is about to finish. So
That's good. You know, the thing is, you're already having a reference. You're going to be having a reference sequence that you can really get, you know, remember the studies on the mutations on various diseases on this area is very important. So if you have a reference genome, then you can go and you can really study a lot of diseases.
Maybe the question I would have about education and use of these genomic studies and how to increase even information of the medical school, health sector, and so on. The people, I think that there is still some gap in the education. What would be your advice? What even like the academia could help maybe in this area?
Absolutely. You know, as I said, all public health sectors have to be informed in terms of the genome and the consequences. All these new concepts now about mental, social behavioral sciences and changes into the genome, people need to be informed. So I think workshops on each country on that will implicate public health public health providers to really educate everybody in this area. It's very important. And what we need is we need to create awareness of all these patterns to everywhere. And we need to be on the same page. This is where we need to go. And, you know, we had, I know your university is doing a lot of in terms of educating and how you can educate the pharmacists, how you can educate the in terms of pharmacogenetics, pharmacogenomics, but the big genome pictures have to be also included in addition just to pharmacogenetics. And it's a very good point that you bring. This is where we need to go. This is the gaps that we need to go as we're moving forward for the public health of the 2030.
I agree with you, and I think with this we can close our webinar. So thank you so much again, Vasilis, for accepting our invitation, for excellent and very thought-provoking presentation. I want to thank also those who made this webinar possible, management of our college and UA University, who gave us green light and supported the organization of the webinar, organizational and technical team, Dr. Azar, Mr. Al-Sajir, Ismail Al-Konaisi, Dr. Ifat, and Ms. Bayan. Thank you also all of you who participated today. I think that you will agree with me that it was very fascinating, very interesting webinar. If you would like to receive certificate of attendance, please fill the link of the survey. It is a very short survey and you will receive automatically the certificate after filling the survey. Our next webinar will be with Dr. Isabel Boden-Løsne from the Norwegian Institute of Public Health, Norway. It will be held on February 22nd, so we will announce soon the details. Please follow us on social media to find more details. We look forward to see you next month. Keep well, stay safe, and goodbye. And again, thank you, Professor Vasilis Vasiliou, for excellent webinar. Thank you so much.
Thank you.
Goodbye.
Bye.